I don't think you usually research childbirth issues.
No, not at all.
So how did you come to this
It was
line of
completely
research?
coincidental. So we were interested in this protein, uncoupling protein 2, and specifically what it does in the brain. And we were doing culture studies when you take the cells in early postnatal animals or around birth, and you culture them and see how they function. And as a sort of control, we wanted to see how during development in utero and ex utero this protein is expressed in this area of the brain. And what we noticed that if the animal was naturally born, the expression level of UCP2 was significantly higher than if the pup was taken through C-section. And that's when the whole thing started to be interesting for us and pursue what this protein might mean for those cells in the long run.
So this uncoupling protein 2 is the protein that's at the center of this research. Can you give the very broad overview of what it is, why it's important, and what role it plays in the brain?
So we believe, and I think many people confirm that the main role of this protein is to enable that your cells use fat as fuel. Most of the time your brain cells would use glucose as substrate to make the central nervous system function, but under certain circumstances they can switch fuel utilization from glucose to fatty acids. And UCP2 is a protein that promotes the utilization and enables the utilization of fat as a source of fuel.
Okay, so can you explain the experimental process you followed?
It was basically very simple. So once we identified that there is a higher expression of this protein in natural birth, then we took the slices and tried to understand what happens to the neurons, what happens to the connection between these neurons if we interfere with the expression of UCP2, either chemically by a compound that inhibits the functionality of UCP2, or by genetic suppression of UCP2 expression. And basically found that the lower the UCP2 expression, the less connectivity develops between neurons of the hippocampal formation. This is part of the brain that is in part responsible for developing memory. It's very important in the learning and memory processing. And then once we established that in vitro setting, then we wanted to know how animals' behavior associated with the hippocampal formation might be different depending on the level of UCP2 expression. And we found that they perform differently in tasks that are associated with the hippocampal function.
Can you explain those differences?
One of the main things that we didn't want to do with this study, which I don't think we would be in the position to make, is to qualify who's better, who's worse. We simply just say that they do behave differentially if they have lower or higher expression of UCP2. And these are, very simple tasks that you put, for example, a mouse in a open field in a novel environment and analyze how they behave. Do they explore novel aspects of the environment, or do they feel more timid and stay around the walls of the new setting? And they show differences, but it's very difficult to say that one is better or worse. Because these tasks are simple tasks and don't really resemble real life situations of even for a mouse, let alone how you interpret the data for the human conditions. All we basically concluded that they do behave differently, and we would like to understand, actually in prospective studies now we are designing, to try to conclude whether for a mouse, for example, in the long run, is it better or worse, and eventually try to find human studies, design human studies where we could, or somebody else could test whether there are differences in C-section versus natural birth associated outcomes.
Do you think those tests are possible? It seems like in humans there are so many other factors.
Of course. I mean, it's very difficult. I take it's very difficult. Mm-hmm. But I think, at least in light of our data and some other publications that will come out, I think in the near future, it's a reasonable question at least to ask, is there any way how this could be tested in humans, considering the high rate of C-sections that are not indicated by medical condition. That's the long-term reason to do these studies.
Okay, because as you said, there are huge numbers of women getting C-sections who don't necessarily need to. Do you see these findings actually making a dent in that trend? Because I know especially in the US,
I don't
it's just
think
an
it
incredibly
will.
high number.
I don't think it will, but I think what I hope it will do is to make people wonder whether it's a good idea to pursue these studies and come to some sort of conclusion what threshold you should reach in order to get a C-section. One of my point was that I was in a Turkish OBGYN society meeting just before the paper came out, and there a lady sitting next to me was a physician scientist, told me that in Turkey, in private practice, which is the majority of the cases for middle class to deliver babies, the rate of C-section is 100%. And that really struck me. And there are many other countries that I think there's a trend to increase to increase the number of C-sections, predominantly due to convenience. And I think if there is a biological process that is making a difference between C-section versus natural birth, I think at least it's just good to know and see how you can put that into your consideration.
Okay, so you mentioned the role of UCP2 in metabolizing fat.
Yes.
And then it's also important in brain development, obviously.
Correct.
How are those roles related?
So we believe that our facts for this are a little bit sketchy, but we believe that these metabolic pathways are very important for your ability to change and adapt to the novel metabolic environment. So in utero, your nutrients are provided by your mother through circulation. As soon as you're out, all of a sudden, everything is changing. You have to take your food through your mouth. It's the breast milk eventually solid food, so that the whole body will have to make an adjustment to the incoming various nutrients. And I think this flexibility in cells to be able to utilize different types of nutrients, specifically during development, where it's important that you build your structures properly and send out projections in the case of the nervous system. I think it's important to, again, provide the cells the adaptability for the new circumstance.
Yeah, do you have any explanations or guesses as to why the protein is induced by natural birth but not by C-section?
So I thought of some during the conversation with other people after the paper came out. I think there are many factors that could play a role. One obvious that you would think of is mechanical pressure, and going through the birth canal is obviously a huge physical pressure on the child. There are obviously hormonal changes that occur during the same period, both from the mother's perspective as well as the child's perspective, and there is the potential for the umbilical cord being suppressed, and the oxygenation of the whole child is altered, so hypoxia could be. There are many factors that I think could lead to or maybe together they lead to the induction of these mechanisms that involves UCP2.
Do you know if there's any research going to be done into the question of why it's induced?
Yes. I mean, we will do it.
Yeah.
Because I'm running a grant right now,
Oh, okay.
October 5th deadline,
Okay.
where we're going to try to play around these things. What are the triggers of these, and actually whether you can mimic it through surgical intervention as well.
Okay, so one of my questions is, what are your next steps in this line of research? So that's obviously one of them. Do you have any more directions you'd like to take this?
Yeah, so we are interested in looking at mouse models and do a more thorough, because this is a coincidental finding. A lot of the lead up to these things were not really properly controlled, so we'd like to have more controlled prospective analysis of what's the outcome of C-section versus natural birth. Potentially doing mouse is a good model for this, and eventually dogs would be good models because they have multiple births at the same time, so you can have some of them come out natural birth, some of them with surgical means, and follow the animals and see how later in their life their behavior is different or not, depending on how they were delivered. That's one of them. Mice, we obviously propose mainly mice studies, but we'd like to explore, as I mentioned, dog studies, and eventually try to see if we can discuss it, which we have started to discuss in the nursing school and here at OBGYN, how we could conceptualize studies in the human population.
Do you think you will be involved in studies in the human population? That seems like, I don't know, so large scale.
I mean, there are obvious proper experts to do those, but for sort of discussing the principles, I think I would be happy to.
Okay. So as we were talking about, their number of women getting C-sections is so large. Was a result like this a concern? Like, was it at all expected that there could be some problems with getting C-sections versus natural birth, or was there a place for
It's
that?
very interesting because after the paper came out, I received a lot of emails from midwives who were very excited about this work. And again, I was not involved before with these issues, but it has been an issue, I'm assuming, for a long time, debate amongst practitioners as well as midwives when and how C-section should be promoted. So it's been there.